So what we're going to do is we're going to pull together several different pieces, mainly having to do with the next steps for the evolution of the land plants, in particular the steps leading up then to the evolution of the angiosperms, the flowering plants, one of the most, well, the most important clade in terms of sheer numbers of species, as well as sheer dominance of current vegetation on the planet. But in order to do so, we're going to have to cover a few things, such as fill in some of our gaps in terms of our understanding of the evolution of leaves, but also understand that the kind of the continued colonization of land actually selected for many different aspects of plants. And one of those important axes of selection actually had to do with plant size, okay? Going from these very small diminutive plants to these massive giants that tend to dominate the biomass and ecosystem fluxes today. But there's an important kind of additional aspect of the story in terms of why natural selection, for one, would favor increasing size, but then also there are all these additional kind of constraints and additional selective pressures then that come with increasing size. One of them being hydraulics, plant hydraulics, and the importance of being able to have a directed vascular network, okay? And so we're going to be talking a little bit more about vascular networks today, in particular, plant hydraulics. And we're going to be trying to wrap all of that up in terms of what we know about these initial radiation of plants and land plants, in particular, this early split in land plant evolution. And I'm going to introduce another topic today. This topic is known as convergent evolution. Now, we've talked about convergent evolution kind of in the past, but we're going to keep bringing up convergent evolution. And convergent evolution is very important because when you find true cases of convergence, phenotypic convergence, that is totally independent clades evolving very similar phenotypes, that is going to be reflecting something fundamental in terms of ultimately why we see the diversity that we see today. Okay. So again, the spermatophytes, this is where we left off with the evolution of the seed, right? And in particular, kind of the key synapomorphy of the spermatophytes is the evolution of the seed itself. Okay. Now there's some additional traits that we've been kind of filling in along the way, but I just wanted to remind you kind of where we are in the phylogenetic tree. Okay. Now I want to go back to this big, important kind of explosion of diversity that happened by the end of the Devonian. So if you remember, we had this major split happening at the end of the Devonian, right? Where we had a line of plants known as the rhineophytes, as well as the Zostrophyllophytes, giving rise to this separate independent line of evolution. But we had on kind of the opposite side, the trimerophytes, and the descendants of the trimerophytes then gave rise then to things like ferns, gymnosperms, angiosperms, seed plants, okay? Now, by the Carboniferous, we ended up last time talking about the Carboniferous. And by the time we get into the Carboniferous, which is between 290 million years ago to 363 million years ago, that's when we start to get a terrestrial planet that starts to resemble kind of modern day vegetation and plants, right? So by the Carboniferous, we actually had really diverse floras. We had a lot of different species, but then we also had forests, okay, trees, okay, big trees, right? And remember this transition coming out of the beginning of the Devonian 400 million years ago into the Carboniferous by 363 million years ago. We went from these diminutive, really small plants to these big honking big trees, okay? In evolutionary terms and geological terms, this is such a quick change, a quick radiation of different phenotypes and traits, in particular plant size. Okay, so a lot of action then happened in the Devonian up to the Carboniferous. Now leaves. Leaves are critically important because they really are one of the pinnacles of plant evolution because they're the engines of photosynthesis. So remember, photosynthesis was effectively kind of restricted to these photosynthetic stems. And if you looked at the total photosynthetic surface area of something like Cooksonia, it was relatively small. The evolution of leaves then enabled this dramatic increase in global photosynthesis. and even the photosynthesis at the level of the individual. So you go from these photosynthetic stems on the individual then to individuals of the leaves. And it's a dramatic increase in terms of the total amount of CO2 then that's assimilated, carbon that's assimilated, then that the plant can use then to grow, perform homeostasis, and then also throw all that carbon into seeds and fruits and reproductive structures, okay? So leaves enabled this huge increase in the ability to build and grow and to do plant metabolism and to do work, okay? And so the evolution of leaf then also had incredibly major global impacts, because if you remember, photosynthesis then involves assimilation of CO2 from the atmosphere and putting that carbon then into the plant body. And this dramatically increased, this structure, the leaf, dramatically increased the ability of plants to assimilate CO2, but then also modify the atmosphere. And as soon as we see the evolution of leaves, we see the decrease, the sucking down of atmospheric CO2 across the entire globe, okay? A major important innovation. Leaves are the engines of photosynthesis, all right? Now remember, out of the Devonian, we had this split, okay? We had the split in terms of these early land plants, remember? So we had this line of evolution. We talk about the rhiniophytes, and then my favorite word, the Zostrophyllophytes. And out of the Zostrophyllophytes, their descendants then gave rise to many different plants, several of them extinct, but in particular the seed plants, all right? So I'm sorry, I read this wrong. This is the Trimerophytes, not the Zostrophyllophytes. If we want my favorite word, we have to go out to the other branch, independent line of evolution, the Zostrophyllophytes, which then gave rise to a series of plants, taxa that are currently extinct, but in particular, most importantly, the lycopods, or the lycophytes, okay, which some of them are still around. Okay, so remember this early distinct line of plant evolution. And we know that actually those two different lines of evolution are associated with independent evolution of leaves, okay? Convergent evolution of a leaf-like structure. One line of evolution, known as the megaphyll line, we call these sometimes true leaves. These are the leaves that you often kind of find associated then with plants today, because all the seed plants, the angiosperms, right? The conifers, right? The rest of your gymnosperms, ferns, these all have megaphylls. Whereas this other line of evolution that took off and was the dominant form of plants, dominant vegetation during then the Cretaceous and back to the Carboniferous, was the microphyll line of evolution. And the microphyll line of evolution, if you want to see these leaves, which are known as microphylls, you have to find lycopods, okay? So the megaphyll line of evolution and the microphyll evolution associated with convergent evolution in terms of this leaf-like structure, okay? So what's the difference then between a megaphyll and a microphyll? All right, well, we can go into a little bit more of those details now. So within the megaphyll line of evolution, what we see are these green photosynthetic stems, kind of like Cooksonia, right? Remember, they have stomates then on the stems, but increasingly during then this period of rapid land plant evolution, we find then fossils with this webbing of lamina. And so a leaf then, a megaphyll leaf, is really then this lamina basically kind of joining together all of these branching structures, right? And so we have this internal branching structure, which is now the venation then of the leaf, kind of reflecting this early kind of evolution based on this non-lamina webbed structure. Okay. And so megaphyll leaves are kind of basically reflecting this photosynthetic lamina that's connecting this vascular branching structure, right? And in terms of their development, these develop together. So as you go from a young bud, then that's developing, then via mitosis to this emerging leaf, the lamina and the vascular tissue kind of develop together, okay? Now we see going from 410 to about 370 million years ago, the evolution of true leaves or megaphyll leaves by about 370 million years ago. So we go from these simple photosynthetic branching structures all the way then to plants will have this infilling between the side branching. So we get this webbing, right, on these side branches. That's a megaphyll. Okay, they develop together. The webbing and the vascular structure develop together when the plant is growing. So it turns out there are many different types of leaves, and I'm going to say leaves in quotations because there's many different ways to have this kind of flat, laminal, kind of big surface area for photosynthesis. But the two major ones are known as microphylls and megaphylls. Okay? So I've emphasized the megaphylls, right, where we have this branch vascular system, right? And these leaves, megaphylls, basically evolved by the fusion then of these branching stems from this webbing of this lamina. Now, on the other hand, a microphyll evolved differently. It's somewhat similar, but developmentally, these are very different leaves. So we have these initial kind of branches then of these early land plants. And so we have the vascular tissue that's kind of on the inside, then in these early vascular plants. And we have then developmentally these sporangia then developing then on the side. Now, selection, it looked like, took some of these initial sporangia, at least some of them, elaborated them, kind of elongated them so that they were no longer sporangia themselves, but instead these more kind of flat photosynthetic type structures. And so the important part of a microphyll is that the vascular system actually grows into it. And I have a few kind of slides and diagrams kind of highlighting this. So within both of these evolutionary lines, the microphyll line, the megaphyll line, we actually see many examples of convergent evolution, right? Independent events then happening kind of throughout then the evolution of the land plants. And so we actually find within the microphyll line many traits similar to the seed plants and evolved independently within the microphyll line. Now, over time, actually, many of these fossil taxa actually went extinct. And instead, what we were left then were the dominant plants that came to basically cover most of the earth, which was the megaphyll line. So when we mention convergent evolution, what does that suggest to you? That sounds like a great midterm exam question. In fact, it's so great, I could even scribble it down right now as a reminder that maybe this will appear on the midterm exam. All right. So within the microphyll line of evolution, remember, this is a distinct line, this distinct lineage of seed plants, okay? And these ultimately gave rise then to the lycopods. Oh, I noticed there's an error here. This is a distinct line of evolution in the land plants, not the seed plants. I hate it when I make a mistake. So please cross that out. That's not seed plant. Please don't be confused. So this is a distinct line of evolution, independent evolution within the land plants. Now, the microphyll line of evolution ultimately gave rise to the lycopods, okay, or the lycophyta. So they share a common ancestor with the Zostrophyllophytes then that are extinct, currently extinct, but their descendants gave rise then to lycopods. Now, these leaf-like extensions, these microphylls, they're another term that we use for them. There's something known as enation, E-N-A-T-I-O-N-S, enations. And these enations are these leaf-like extensions, okay? So an enation is a microphyll. And enations enables you to increase photosynthetic surface areas. And a good way to think of an enation is like a hangnail you get on your thumb or one of your fingers, right? Totally annoying, right? Drive me crazy. But an enation is kind of like a hangnail off this main stem, okay? So an enation equals a microphyll, all right? So what's horrible about botany is that we often have multiple words all kind of meaning the same thing. So microphyll is an enation. All right, so it's not the same as a true leaf, but functionally very similar. It increases photosynthetic surface area. It kind of has a lamina, but developmentally very different. And some of these in the fossil record actually reached about 78 centimeters long. Okay, really big. So enations in the fossil record, actually we have records of them getting quite big. So not all leaves are the same, all right? So enations equal microphylls. These are these scaly kind of hangnail, kind of leaf-like extensions, all right? And this is an independent evolution of leaf-like structures. So what I'm gonna do now is I'm gonna list out several different cases of independent evolution, convergent evolution in the microphyll line of evolution, okay? But before I go into kind of listing out all those details, I wanted to kind of go into a little bit of detail in terms of how annations or microfills are different from megaphills. Okay, so an annation is kind of reflected in these diagrams. So here we have these kind of early kind of microfill taxa where they have this central stem, and we have the term here is called prostele. Don't memorize that. Okay, this is another botanical term. This is the vascular tissue, the internal, these tubes. Remember, we have these tracheids, right? Because these are all vascular plants. And so we have these internal vascular system. And so as the plant is then growing and developing, and we can catalog this by the fossil record, these annations develop as these kind of these photosynthetic kind of extensions off the side, kind of like your hangnail, right? It's like this little flap or this little scale then that develops. But as it then develops, notice we have the lamina developing completely independently of the internal vascular system. But then as the annation grows, then we have the vascular tissue starting to grow and then grow into it, okay? So your venation network actually grows as it develops into the annation itself. This is completely different to how a megaphyll develops, where the vascular system and the lamina develop together, okay? So the fact that the vascular system grows into it is developmentally totally separate, okay? And you can see that again here. We have our green photosynthetic stem, our red vascular system, and then as the annation develops, the photosynthetic scale or the lamina develops first, and then this leaf trace or the vascular system then grows into it. So totally different, a different way of building effectively the same structure, all right? So the end result is that we have a leaf-like structure, and the function is the same, right? The function of a leaf is to increase the photosynthetic surface area. But evolution has found different ways then to develop then that structure, okay? So in the end, convergent evolution of a leaf-like structure, but different developmental ways of achieving then ultimately that goal. Pretty cool. Okay, so within the microphyll line of evolution, we have the convergence of a leaf-like structure. Now, we also find within the microphyll evolution, the independent evolution of roots, okay? These root-like structures with the same function of extracting water and nutrients and having a vascular system to then transport that to the rest of the plant. We're going to be talking more about roots in a little later. But we see within these two separate lines of evolution, this convergent evolution of several different structures, leaves, roots, okay? We also see the independent evolution of something known as the vascular cambium and secondary growth. We're going to be talking about the vascular cambium and secondary growth several lectures from now. But the vascular cambium and what's known as secondary growth enables stems, a cross-section of a stem, to increase in size, right? So as you go from a small little sapling of a tree to a big tree, you have to be able to grow in girth, grow the stem in girth on the outside. And so we see that, we see the ability then developmentally of the ability to increase then in girth of the stem, okay? And they achieved it in a totally different way. It's pretty cool because within the vascular cambium, within the microphylls, those vascular cambium cells in the stem actually didn't undergo a type of cell division known as radial longitudinal division. That's a really kind of technical kind of term, and it's a mouthful, but basically that is a different way of splitting cells than as they grow. And it actually had a big functional implication because it turns out you can't do radial longitudinal division for long within the stem because you ultimately kind of reach a maximum size, kind of limits how big you can get. And that was probably one of the reasons why the microphyll line of evolution was ultimately almost demised and ultimately brought to extinction by the rise then of the megaphylls. Okay, and so as a result of this, really, you can't really get that big. Okay, there's a limit on how big you can then grow. All right. So within the microphyll line of evolution, convergent evolution of leaves, roots, and secondary growth, the ability to get big in the stem. All right. But what's cool is that as through time, as paleontologists started to discover more and more of these fossils and pull together and piece together how then these plants were built and then how they grew, we actually discovered that there were several different cases of independent evolution, in particular, the independent evolution of heterospory. So we see within the microphyls, we start to see plants within that separate line of evolution, that separate lineage, having then descendants then that evolved the capacity to do heterospory, which then passed along then that trait to their offspring. Same thing in the megaphils. We see the evolution of heterospory, and then those gave rise then to descendants and passed along that trait. Convergent evolution. And there's even some indication within the microphyll that the megaspore in some species started to be retained within the sporophyll. There's a little bit of debate about this currently, but that seems to be very similar to the ovules in seed plants, right? So this case was they're not true ovules, but it was kind of ovule-like, right? And so in some ways, this is almost similar to the evolution of seed plants, almost convergent evolution of seed plants, but very close. Now here, the sperm still needed to swim. Okay, there's still the restriction, tight restriction in growing in really wet areas. And what's also cool is the evolution of the tree-like habit. All right, and so we see the evolution of big things within microphyll and the evolution of big things within the megaphylls. All right, so 6 different cases of convergent evolution. And I'll walk through them really quick. Leaves, convergent evolution of leaves, but these leaves are known as microphylls or innations, developmentally very different. We see the independent evolution of true roots. We see the independent evolution of the ability then to get big via a vascular cambium. But this vascular cambium was a little different in terms of how it was arranged, and because of which that placed a limit on how big microphyll taxa then could get. Independent evolution of heterospory. Independent evolution, we think of the megaspore in some species being retained in the sporophyll, all right? Kind of like a pseudo-ovule, right? Again, very similar to ovules in seed plants. Again, sperm still needed to swim. And then lastly, we see the independent evolution of the tree-like habit, okay? All right, so I just outlined several cases of convergent evolution. We're going to be coming back to convergent evolution, but that kind of sets the stage, kind of describing this explosive radiation of land plants in both the microphyll line and the megaphyll line. And as this was going on, this incredible radiation of the land plants coming out of the Devonian, there were other huge implications for the entire biosphere and as well as the entire planet. So it turns out that the evolution of leaves in particular and the evolution of increasingly larger plants had tremendous atmospheric and ecosystem consequences. Okay? And because of which that influenced their further evolution as well as the evolution of animals on land. So one of the most kind of important signatures of the rise of the land plants was that as you increasingly colonize more of the planet, that's a lot of leaves, that's a lot of chlorophyll, that's a lot of rubisco on the planet. And that's a lot of assimilation of CO2 that's happening. And so what I'm showing you here, this is a complicated little graph, but it's a recent, relatively recent compilation of several different ways of reconstructing atmospheric carbon dioxide, going back to about 500 million years ago. Okay. So with the rise of the land plants, photosynthesis, remember, assimilates CO2 out of the atmosphere and puts then that CO2 into the plant body. And I've already told you that a lot of those plants growing in these wet, swampy environments deposited a lot of that carbon, what's now known as fossil fuels. Okay. So if you notice, we have several different lines here. These are different ways of estimating atmospheric carbon dioxide. Actually, one of them, something known as the Rothman method and technique, it actually does a pretty bad job. And now we've effectively ruled out this pink model because there's other ways now that scientists have come up with that we can test and verify, these are our best estimates in terms of atmospheric carbon dioxide. And so if we go back to about 500 million years ago, before the evolution of the land plants, atmospheric CO2 was quite high. But if you notice, around 400 million years ago, when the land plants effectively evolve, we see this big drop in terms of atmospheric CO2, okay? Now, atmospheric CO2 kind of bopped around for a little bit, and there was actually quite a bit of volcanic activity, okay, at different parts that likely also increased CO2 levels. But by the time we get to about 100 million years ago, approximately, this is when the angiosperms evolved. And this is where basically the vast majority of the planet started to be covered basically with leaves, okay? And after the evolution of the angiosperms, they further then dragged down atmospheric CO2 concentrations. And so if you notice, there's a huge reduction since the evolution of the land plants in terms of atmospheric CO2. And if you're confused on this graph, remember this goes back in time and present day is current right here. Okay, so this is an important context. So it looks as if plants had a tremendous impact on our atmospheric CO2. All right, now I also want to point out that during this time, there were two major extinction events, something known as the Permian Triassic extinction event that occurred about 252 million years ago, and something known as the Cretaceous Paleogene, or the KPG extinction event 65 million years ago. So this KPG event 65 million years ago, this is the asteroid that took out the dinosaurs. The Permian Triassic extinction event also looks to be a very large asteroid as well. Two kind of rare events, big asteroids hitting the planet, basically decimating a lot of life. The Permian Triassic one was pretty devastating. About 90% of animals that were around went extinct. But what's actually really interesting is that there are barely any plant extinction events recorded here. There's a good likelihood that a lot of plants were kind of killed and everything, but in terms of the species and our fossil kind of evidence, we don't see really big extinction events happening. But what did happen was that the dominance changed. And at this time, associated with this big extinction event, the gymnosperms replaced the lycopods in terms of the dominant taxa. That seems to maybe have been associated with this big external disturbance event. Now, the Cretaceous Paleogene event, that led to the extinction of about 75% of animals. But what's interesting is that only about 57% of plant species became extinct. So plants are a little bit more resilient. Now, what we find immediately afterwards were that ferns rapidly colonized after this big extinction event. And also during this time, fungi started to dominate them as well. And we see this shift from dominant evergreen species to deciduous species. And we'll talk a little bit about what it means to differ between evergreen and deciduousness, but this is the ability to withstand long periods of seasonality, cold or drought, by dropping your leaves, right? So the KPG extinction event looks to have been a big selective event that basically the species or the taxa that made it through were the ones that had the ability to be deciduous, to have the ability to withstand kind of major kind of stressful events. Okay. We'll come back to this in a little bit. All right. So remember, we have, coming out of the Devonian, a lot of this radiation then of the land plants. But the one thing I want to return to right now is a very important component. As we go from the Devonian into the Carboniferous, we see a dramatic increase in plant size. Okay. And I want to talk a little bit about the importance of plant size, because we kind of take for granted the fact that we have big trees kind of around. And the evolution of being a big plant, a big tree, is actually not simple. Okay. It takes a lot of different evolutionary steps in order to get big. But it's clear that there has been important selection, persistent selection for increases in sizes in certain environments. Okay, we still have a lot of small plants around, but certain environments select for increases in big size. So I want to show you just a few of the dramatic kind of examples of differences in size. If you want to go within the seed plants, and in particular within the angiosperms, some of the smallest seed plants around, something known as either wolfia or actually duckweed. Okay, duckweed is lemna. Sometimes you'll go to ponds, often ponds where you see swans and ducks around, and you see this kind of green slimy kind of surface on the top. Well, it's actually not a green slimy surface. This is actually a spermatophyte, and in this case, angiosperms. Each individual exceptionally small. You have these little roots, you have these little leaf-like structures, right? And you even have little flowers and little seeds as well, right? Incredibly small. But you compare that then with some of the largest seed plants, in particular, some of these large gymnosperms, such as the sequoias, okay? And so to this day, this is actually the biggest, most massive organism then on the planet. It's actually an order of magnitude bigger than any of the most massive dinosaurs or even the current largest animals on Earth, which are the blue whales, an order of magnitude larger in terms of mass. So these things live about well over 1,000 years, some approaching almost 2,000 years old. Okay, these are not only massive, but they're also ancient. Okay, and they're also quite tall, approaching about a little over about 120 meters in height. And so tree height is actually interesting because obviously if you're a really big tree, you have to transport all of your resources from the soil through the roots all the way up to the tippy top, to the leaves all the way up to the top. So it turns out that being able to transport resources from the soil all the way up to these enormous kind of leaves, kind of at the tippy top, was not a simple feat. Okay, we all love big trees, right? And unfortunately, some people like to put tunnels through them to show how big they are. But the open question is, well, why then selection for increased size? Okay, why get bigger, right? Why these massive structures? Especially if you think of the early land plants when there are these little diminutive things, right? What's the selective advantage of being really big? Now, what's really fun kind of in botanical circles, if you really want to start a fight with an Australian botanist, you tell them that the biggest trees around, the tallest trees around are the sequoias and the redwoods in particular in California, right? Because we all know they're the tallest. Well, it turns out that the Australians think that their eucalypts are some of the tallest. And it actually turns out that if you look at some of these kind of fossilized eucalyptus and some of the early reports before people started cutting these things down, well, it could be that some of these eucalyptus trees growing in southern Australia into Tasmania may actually have been bigger and taller than some of these redwoods. Okay. And it actually turns out that if you look in different parts of the planet, if you look at some of the amazing forests in Borneo and Indonesia, and even some of the really tall rainforests kind of deep in the Amazon, you can find tropical trees that actually start to approach a similar height of about 100 meters, okay? So in multiple parts then of the planet, in both gymnosperms and angiosperms, we see trees approaching these enormous heights, 100 meters, which is really incredible. All right, so just within the seed plants, we see selection for increasingly small size and increasingly larger size. Enormous differences in size. So over time, within each kind of major land plant clade, we see the increasing evolution of taxa that are increasingly larger and larger. Now, this is known as something known as Cope's rule. And Cope was a paleontologist. He was actually a zoologist in the late 1800s. And he noted that within animals, if you track then the evolution within each independent clade, there was a tendency within that clade for taxa to become larger than the common ancestor, okay? And we see that repeatedly in the fossil record in both animals, but also within plants. And so if we plot the record stem diameter as a function of geological time from 400 million years ago to present day, what we actually see is the progressive steady increase, and by the time of the angiosperms, a rapid increase in terms of record stem diameters and record heights. Okay, so there's been this progressive selection in certain environments for taller and taller and bigger individuals. And so my open question to you is, what are the selective pressures in certain environments for increasing size? Why get big? Okay, so that's my open question to you. And so I want you to think about that, because we're going to be returning to that, and it's going to be kind of an important component, because if there is consistent evolution for increases in size in certain environments, that also brings up some very important physical constraints in terms of the evolution of the functioning of land plants. Okay, so for the continued colonization of land, that required specific adaptations for actually many different functions. Okay, so in order for plants to colonize these increasingly drier environments, we also needed selection for an efficient internal transport network, as well as increasing selection, continued selection for desiccation resistance and avoidance, as well as reproduction without water, but also effective dispersal, as well as the importance of competition. In particular, increased adaptations for dealing with competition. Because as the planet became basically filled up, all of land became filled up then with plants, plants started then to increasingly compete with each other for similar and also for multiple resources. Okay, so in the context of this, what enabled plants then to continue to increase in size? So this is where we start focusing on what are the kind of the main general kind of principles then influencing whether or not plants can increase in their size. And this is where we start to talk about the importance of plant hydraulics and gas exchange. And it turns out that if there's selection to become taller or bigger, that comes at a detriment, at a cost in terms of your ability to transport resources to increasingly larger heights. So if you remember, Fick's law tells us that passive diffusion is very slow, okay? And so if plants relied exclusively on Fick's law, it would take a long time, increasingly longer times, than to transport then resources, nutrients, water, to increasing heights, okay? And there's also something fundamental that also shaped the evolution of plants. And that is something known as the photosynthesis transpiration compromise. You know what photosynthesis is? That's the exchange then of CO2, all right, from the atmosphere to the inside of the plant in order then to kind of make sugar. Transpiration is also the exchange of gases, but the exchange then of water. So there's this compromise between the uptake of carbon and the release and utilization of water, okay? So pipes. The vascular plants are characterized by the synapomorphy of a pipe, a trachid, okay? And we're gonna delve into some details of what trachids are in just a little bit. But there's gonna be another pipe involved, and that pipe is known as the vessel, okay? So basically, land plants, vascular plants, have two types of pipes. It's either a vessel or it's a trachid, okay? The synapomorphy for all vascular plants is the trachid, so the trachid came first. Now, throughout the evolution of the tracheophytes, the vascular plants, if we go back to the Silurian, the lower Devonian, up to the upper Devonian, if we actually look at the trachids within fossils and we measure the diameter as well as the length of these different pipes, because these actually fossilize really well. You can crack open rocks and you can see these fossilized trachids really well, and you can measure these in the lab. And so as you go from the Silurian up to the upper Devonian, what we actually see is this tremendous increase in the radius or the diameter of the trachid. Basically, what we're seeing here, as you go from the upper Silurian and the lower Devonian to the upper Devonian, you go from small pipes to big pipes, okay? Interesting, because that's telling us something very, very important. So also during this time, as we go from the Upper Silurian to the end of the Devonian, we see this rapid, incredible evolution of forests, going from basically something like Cooksonia, which is a diminutive little dinky thing. By the time we get to the Upper Devonian, we can see these tall plants and forests. Also along that time, we see this big change in terms of the sizes of pipes. Okay. Now you may be thinking, my gosh, this is the most incredibly boring thing you've ever been teaching us about, but this is really, really cool. And so it reflects fundamental aspects of how physical processes have shaped plant evolution, but also how natural selection has dealt with that physical constraint to come up with an ingenious solution to transport resources to increasing heights. Okay. So we need to kind of go back to first principles. I'm showing you an equation here. This is something known as the Hagen-Poiseuille equation. Okay, the Hagen-Poiseuille equation. And so I'll kind of walk through this. Okay. On the left side is the equation. On the right side is this little diagram. And so let's go through this diagram because this is going to help. So what this is a pipe. Okay, literally this is a pipe. And you know, if you're take engineering courses and you're into hydraulics and everything, gosh, this is what they teach you. But we're just going to take a little bit of some engineering principles here. So here's our pipe, and we can characterize its radius as well as its diameter. And that pipe is going to have a length, right? So here's radius, and here's the length. Okay. Now imagine fluid moving then through this pipe. Okay. As fluid is moving then through that pipe, these different arrows represent the velocity profile of fluid moving through. If you're near the center of the pipe, the speed at which fluid is moving is fast, but then as you move then out to the surfaces, the speed slows down. And it slows down, remember this is related to the boundary layer we talked about. It slows down because there's this friction, right? So as fluid is then moving across then the surface, it kind of slows down, right? So fluid then that's close to the surface then of the pipe is going to be moving slower than fluid then near the center of the pipe. Gosh, it's just hydraulics. Okay. Now, the fluid flow rate, how fast fluid is moving then through the pipe, is going to be equal to this simple ratio. And in the numerator then of the ratio, we have R, which is the radius. And notice the fluid flow rate is then going to be equal to how big the pipe is, its radius. So as we increase the radius, as we increase the numerator, the fluid flow rate will increase. But look at this. We had this big honking exponent here of a fourth power. That means that there's a fourth power dependency of flow rate on the radius. So if you double then the radius, you have to actually take the fourth power then of that change, right? So that is a huge explosive increase in terms of the fluid flow rate. And so what we can take away from this is basically tubes then that are bigger in terms of its radius conduct fluid at a much faster rate. But now what I want to emphasize to you is that in the denominator, we have 8 times nu. PowerPoint failure. That should be nu, not a question mark here. That's the fluid viscosity. Okay, nu is the fluid viscosity. Let's assume that that's constant. Let's not bother with that. Okay, so that's the fluid viscosity, but then we multiply that then by L, which is the length. So if you have then a plant where the radius of the tube, radius of the trachea is the same, it doesn't change. We imagine a plant built to tubes the same size, but then we have selection that increases its length, right, or its height, right? And so these pipes then have to supply increasingly greater heights. As plant length increases or the length then of the tube increases, notice because it's in the denominator, that means that the fluid flow rate has to decrease. So the bigger the transport distance, the lower the fluid flow rate in the end because of the increase of all of this resistance through the surfaces then of this pipe, okay? So whenever I show you then an equation, the first thing I'm going to ask you is, what variables can natural selection operate on? So on the one hand, natural selection is operating for plants to become bigger, taller. We see that in the fossil record. But with that comes this trade-off in terms of fluid flow rates. Now, it turns out that given the Hagen-Poiseuille equation, natural selection can also operate on the radius then of these tubes. And that's apparently what we see in the fossil record. With increases in plant height, we also see a concomitant increase in the radius then of these tubes in order then to keep fluid flow rate kind of similar or maximized. Now there's one more term here, something known as delta psi. Again, I have a PowerPoint failure here. That should be delta psi. That's the pressure gradient between the soil and the atmosphere. We're going to come back to this when we talk about water potentials. Okay, so we're going to bookend this right here, and we're going to kind of, put a pin in it. We're going to leave it there, and we're going to come back to this. So engineering principles are important for us to understand why not only selection then for increased height influenced the evolution of the conducting kind of material or the conducting tissue itself. All right, so I also want to introduce to you two different terms here because this can be confusing. We're going to be talking about resistance, hydraulic resistance, as well as hydraulic conductance. Resistance is the inverse of conductance. So conductance we often talk about in terms of K, but resistance is the inverse, we call that Z. Okay, so the total resistance then of fluid then moving through pipes is equal to how tall or how length then, how big in length then that pipe is or the plant is, divided then by the average radius then of the pipe, whereas the conductance is the radius then divided then by the length. They're just the inverse of each other. So increased resistance decreases then the fluid conductance then of a pipe. All right, so increased resistance reduces flow of water to the photosynthesizing tissue. All right, so that's the Hagen-Poiseuille equation. So again, we can go through then the exercise. What happens to resistance with changes in length versus changes in radius, right? So resistance decreases linearly, okay, with decreases in length, okay? So as the length then decreases, your resistance then is going to decrease. If your length increases, right, your resistance increases as well. But your total resistance is going to decrease with the radius, but to the fourth power, okay? And in this case, the -4th power. So as we go then and we study the evolution then of the vascular plants, we see within the vascular plants the evolution of plants then that have something known as the vessel. Vessels are huge tubes, okay? And vessels are characteristic of the angiosperms, whereas within the gymnosperms, they're largely restricted to have tracheids. Tracheids tend to be relatively small tubes. They can vary in their size, but once we get to the evolution of the vessel, that enabled the vascular plants to have these huge tubes, which then had dramatic differences in terms of their lower resistance and their increase in their conductance. Okay. So throughout the evolution of the tracheophytes, we see evidence of selection to minimize hydraulic resistance to transport. And that likely enabled plants to increasingly obtain larger and larger heights or larger and larger sizes. Okay. Now, that's the hydraulic aspects of increases in size. Now, there's this other constraint having to do with this photosynthesis transpiration compromise. And so on land, plants require, and I think I emphasized to you earlier in class, but this is another point to kind of stop and emphasize this. On average, plants require about 138, almost 140 moles of water loss per 1 mole of CO2, and that's assimilated by photosynthesis. So plants have to blow off a lot of water in order to assimilate just 1 mole of CO2. Okay, but remember, as plants then, as selection then has increasingly favored plants then to invade increasingly drier environments, remember, water is going to be more limiting. Okay, and so the luxury of being able to blow off all this water in order to assimilate CO2 is going to be increasingly limiting. All right, so increased transpiration rates are great because that increases photosynthetic rates, right? And so if there's selection then for increases in the ability to get bigger, okay, increase your photosynthetic rate and take all that carbon and grow this big structure, right? We have this other constraint having to do with this photosynthesis transpiration compromise, okay? So increased transpiration, increased photosynthesis, decreased transpiration, decreased photosynthesis. And remember, transpiration is your water use, okay? It's how much water is used in the process of photosynthesis. So this is the photosynthesis transpiration compromise. And effectively, land plants haven't been able to really get over this constraint. In order to assimilate CO2, they have to blow off a lot of water. Okay. And so that gets us to the rise of the angiosperms, okay, the flowering plants, right? Effectively, if you randomly went outside and you touch the first plant that you find, in all good chances, it's going to be an angiosperm, a flowering plant. So the seeds of angiosperms actually develop within the protection of fruit. And so we haven't talked about fruit yet. And so fruit is really cool, all right? So we've talked about seeds, but we haven't talked about fruit. And what's amazing is that actually within the angiosperms, the archegonia is gone. Oh, it was reduced to such a small size that basically it was diminished enough and it's effectively gone. Poor archegonia, oh my gosh. We've spent so much time, right, talking about archegonia is the synapomorphy of the land plants. Yes, it's the synapomorphy because all land plants share that common ancestor. That common ancestor of all land plants had the archegonia, but within the angiosperms, it was lost. Oh,
Oh, well, actually, can I add something quickly?
sure.
I actually learned that in another angiosperm seagrass, their stomata have actually atrophied.
Yeah, it's kind of crazy. Okay, so keep that thought. We're going to come back to seagrasses in a little bit. Okay, now fruits. Okay, we haven't talked about fruits. Fruits, a major innovation of the angiosperms. And basically all of what you're seeing there when you think about fruits are angiosperms. Okay, so what is the fruit, right? Again, we talked about seeds and we talked about sporangia, talked about different reproductive structures. We haven't talked about fruits. What are fruits? So the angiosperms are the flowering plants. Amazing group of plants. Oh my goodness. All right. Basically dominating the species diversity of planets on the planet in terms of plants are angiosperms. Angiosperms are the dominant clade of plants over most of the earth. If you randomly dropped you down someplace on the planet and you randomly went around touching plants, most likely you're going to run into angiosperms. Angiosperms, once they arose, they basically pushed out the dominance of most of the other clades on the planet. They were a superior competitor for a number of reasons. Now, there are certain environments where actually angiosperms don't do as well. And in those environments, ferns, gymnosperms, and even bryophytes can dominate, okay? But increasingly, those are very small, rare parts of the planet where those other clades tend to dominate. All right, so the first angiosperm likely evolved around 120 million years ago, about 125 million years ago in the Cretaceous, okay? Relatively late, okay? So most of the land plant evolution, it actually turns out from about 400 million years ago to 125 million years ago, dominated then by bryophytes, pteridophytes, gymnosperms. All right, so the vast majority of life on Earth have been dominated not by the angiosperms, but by these other groups of plants. Angiosperms are relatively new in the grand scheme of things. Only 125 million years then we've had angiosperms, but as soon as they evolved, they had the critical traits that enabled them to not only outcompete what came before, all the lineages that came before. They had superior competitive traits, but they also had the traits that enable them to colonize the driest and the coldest parts of the planet. Okay. Our first likely angiosperm is something known as Archaeophractus by about 140 million years ago. So a little older than what I said. So by 125 million years ago, we definitely had angiosperms. Our first fossils started coming around 140 million years ago, the early Cretaceous. And so here we have, you notice this is a megaphyll. So we have true leaves, okay, green photosynthetic stems most likely. But then we have these reproductive structures. And we have these leaf-like reproductive structures that were kind of folded, right? And so you kind of see these folded leaves right here. And these folded leaves actually contained on the inside the ovule. And so you can kind of look kind of closely on some of these. And you actually can reconstruct, and you have good structures associated with ovules. And so these are leaves then that are beginning then to contain ovules. Folded leaves with ovules on the inside. This is the origin of something known as the carpel. Okay, the carpel is a key innovation of the angiosperms. So when you think about the flowering plants or the angiosperms, you immediately think of, well, the flower, right? All right, so let's go through and do some definitions. Okay, so a flower is a determinate shoot bearing sterile and reproductive appendages. Effectively, the flower developmentally is a bunch of leaves, okay? Shoots and on the shoot is a stem bearing leaves. So we're gonna have stems and leaves. The flower developmentally is modified stem and leaves, okay? So a flower is a determinate shoot bearing sterile and reproductive then appendages. Flower parts are all highly modified leaves. Although flower parts don't look like leaves, okay, developmentally, they are leaves. And the flower consists of several different parts: sepals, petals, stamens, and carpels, okay? Now, you've likely have had some bits of this maybe in introductory biology. Okay, so we're going to memorize some of these important parts because there is an important evolutionary kind of component to the evolution of the flowers. So the flower technically consists of sepals, petals, stamens, and carpels. There's going to be a little bit of exceptions here, but in general, that's what a flower is. Okay, now key definitions. So I want you to memorize these. Okay, now stamens are structures that are going to be bearing or kind of containing the microsporangia. Now we call these within the angiosperms anthers. Okay, so stamens bear microsporangia. All right, the pollen grains are then going to develop from the pollen microspores, just like in the gymnosperms. But we have these additional structures known as stamens. Stamens then are bearing the microsporangia or the anthers, and they're going to be then responsible for the development of pollen grains or these microspores. Now, here's a key definition. And it turns out then that a key synapomorphy of the angiosperms is something known as the carpel. And the carpel is a modified angiosperm leaf then that contains or bears on the inside of this folded leaf one or more integumented megasporangia, which is an ovule. Oh my gosh, all these botanical terms, they get so confusing, right? That's why I'm emphasizing it's important just to memorize these. If you memorize them, if you know that an ovule is just an integumented megasporangium, this makes sense. But a carpel is a modified leaf that's starting to wrap over and contain these integumented megasporangia or these ovules on the inside. All right, and this is where the second integument comes in. The carpel is the second integument. If you remember the gymnosperms, we only talked about one integument, which is the ovule. The ovule just contains one integument. But within the angiosperms, it not only has, you have the ovule with the one integument, but now we have this other leaf-like structure that's kind of enclosing the ovule to give it its second integument. The carpel makes the second integument. And because the carpels surround these ovules are no longer naked, as in the gymnosperms. Instead, the ovules are hidden from view. And if you parse out the term angiosperm, it literally means hidden seed. Hide the seeds from view. Seeds are contained within the carpel. Wonderful, beautiful. They're no longer naked, but they're contained then within the carpel itself. Okay, angiosperms, seeds are hidden, seeds are contained. All right, so memorize the carpel. The carpel is a modified angiosperm leaf that is then wrapping around the inside integumented megasporangia, or the ovules, giving then the ovule the second integument. Okay, gymnosperms have one integument, angiosperms have two. They have two because they have the carpel. Okay, so the carpel is this modified angiosperm bearing leaf. And so here we see from the fossil reconstructions, we can see on the inside, here's where the ovules are. And we can then see fossils then that contain this leaf that progressively over time, selection then has provided this increasing additional maternal protection, additional kind of surrounding and sheathing layer, kind of burying then the ovules on the inside of mom. Okay, so we have this additional maternal structure and maternal potential and maternal investment around it. And this additional maternal tissue layer is going to be very important for the development of fruit. Okay, carpel, evolution of the carpel. We see then ovules on the outside or on the inside even of megaphils. Okay, so we have this leaf-like structure, and then we see ovules then kind of associated with it. But over time, we see then this kind of folding over then of this leaf. So then ultimately, we get more protection of the ovules inside of maternal tissue, so that ultimately we're left with this structure known as a carpel, a folded over leaf with the ovules on the inside. Okay, pretty cool. So the carpel was an ingenious innovation of the angiosperms and enabled them then to have selection for a number of elaborated carpels and other things that you could do with a carpel. Okay. So what are angiosperms? Angiosperms are flowering plants, all right, and they're tremendous in their diversity. We rely on them every day for our food, all right, for beauty, right, and for just our enjoyment of nature. Angiosperms are flowering plants, but they're also fruiting plants, okay? Because fruits, flowers are all centered around the carpel, the evolution of the carpel. Angiosperms also are perhaps the most they are the most dominant clade on the planet in terms of pure vegetation, but also in terms of species. And so if you look at the total number of species of angiosperms, there are different estimates, but approximately 257,000 species of angiosperms on the planet. Compare that with the dinky gymnosperms. Look at that. There's only 820 species of gymnosperms on the planet. There's about 12,000 species of ferns. Ferns are pretty diverse. About 1,000 lycopods still, from only 5 different genera. 9,000 liverworts. Mosses are pretty diverse, it turns out. Mosses are still pretty diverse. So in general, the bryophytes are still impressive in terms of their diversity. But look at what happened to the poor gymnosperms and the poor lycophytes. Basically, with the rise of the angiosperms outcompeted gymnosperms, they outcompeted the lycophytes, so that they're only restricted to these small, kind of poor resource environments. Okay. Enormous diversity. And they have enormous diversity of growth form and habit. Not only can we talk about trees, right, but we can also talk about these crazy plants that we have in Tucson, right? We have saguaros, we have ocotillos, we have various other cactus, right? Crazy growth forms of desert shrubs and succulents. You have these crazy alpine cushion plants. There are even parasites, right? There are even angiosperms that are parasites that are effectively not photosynthetic and parasitize other plants for their photosynthate. So we have these angiosperm alpine tundra shrubs. There's even aquatic angiosperms that have actually returned back to the fresh water and also seawater. We have epiphytes, and we have crazy growth forms, plants known as lianas. Lianas are woody vines. And if you go to really wet aseasonal forest, especially tropical forest, you can see these amazing lianas, huge, huge woody vines, basically structural parasites. But then once we get into the angiosperms, we have this explosion of different means of pollination, right? We don't really see that within the gymnosperms. We see a little bit of coevolution in the gymnosperms between animals and plants in terms of dispersal. I told you about these juniper berries and yew berries, right? Berry is a horrible word. But within then the angiosperms, this is where we see this incredible explosion of the diversity of pollination methods. Pollination by insects, pollination by wind, pollination by birds, pollination by mammals of all things. Oh my goodness. Diversity of dispersal types, right? And so not only can birds and mammals and also kind of insects disperse as well as wind, but there's even cases, in fact, increasingly, if you go into the Pantanal of Brazil, we're now actually discovering that fish are a critical dispersal agents of some of these seasonally flooded areas in some of these seasonally flooded tropical regions. And so in the Pantanal, incredible place. It's probably one of the last huge pristine wilderness areas on the planet because it gets flooded, right? So it's not open to agriculture a lot. And within the Pantanal, they get incredible jaguars and things. And so because it's been flooded, it's really hard to work in here. And now within the last decade or so, people have gone in and actually discovered that they couldn't figure out who was dispersing a lot of these different species of plants there. And it was only these crazy botanists that actually decided to stay there when all the big flooding events happened, that they discovered that all of these trees were dispersing then their fruits in the flooded time. And once the fruits actually land into the water, these huge fish, these giant tambaquis would actually eat then these seeds, and they'd swim up and down parts of the Amazon, the tributaries, and they would then deposit then the seeds out. And it turns out that a lot of the species within the Pantanal and other flooded areas actually rely on fish for dispersal. Crazy stuff, right? So the angiosperms are now the dominant clade of plants over most of the earth. So we have grasslands, evolution of grasslands, right? We haven't talked about grasslands before. Grasslands here in South Africa, these amazing African grasslands. Deserts, we're blessed to live in the Sonoran Desert. Very unique kind of area of the planet, right? Dominated by angiosperms. Agriculture, oh my goodness, all of our agriculture, the evolution of humanity, owes its entire existence to angiosperms. So rice paddies here in Southeast Asia, angiosperm. Wheat, an angiosperm, right? Everything that you see there, fruits, those are all angiosperms. So why are angiosperms so dominant and diverse? Okay, so I told you this, the evolution of the carpel, key synapomorphy. But it turns out that the evolution of the angiosperms involved this whole suite of additional traits that enabled them, basically the Swiss Army kit, a toolbox by which selection could operate on that allowed them to diversify and dominate then the planet. All right, so why are angiosperms so dominant and diverse? So as we move forward here, I'm going to be asking you then some questions. And these questions then are going to likely appear on the midterm exam. But I would like you to use these questions to bring in what you know from the previous lectures. So what are the key evolutionary innovations of the angiosperms? We've touched on a little bit. What are the innovations then that favor their dominance and the abundance and a biomass of the angiosperms over much of the planet? So where are the angiosperms not the dominant group of land plants, and why? Okay, so one of your readings here coming up is this Waring 2002 paper. But then also, which innovations might have favored the diversity and the evolution of so many species of angiosperms, and how then do you think that operated? Okay, so we're going to be exploring then these questions, but I'm giving you these questions because I would like you in your studying, and hopefully you're forming some study groups, reach out to folks in class, work through these questions, okay? Because these are somewhat open, kind of open questions, right? And I want you to draw on the information that you have to best kind of answer these questions. All right, so it turns out, and I'm going to kind of end here. These are going to be my last slides here. And we're going to kind of follow up on this in the next kind of lecture. But the key angiosperm evolutionary innovations turned out to be kind of four main things. One is something known as double fertilization. And it's exactly what it sounds like. Double fertilization, 2 fertilization events. Crazy, kind of weird. We're going to fill in the blanks on what exactly double fertilization is. But it turns out double fertilization is really key because double fertilization enabled the provisioning of the seed with a different kind of flavor of nutrients, okay? Double fertilization. The carpel, we already went over the evolution of the carpel. Carpel was a key evolutionary step, basically the synapomorphy then of the angiosperms. But also the flower, the structure of the flower itself, as well as fruit. Okay, so key evolutionary innovations of the angiosperms, double fertilization, carpel, flower, and fruit. Now, if I ask you for the key synapomorphy of the angiosperms, definitely give the carpel. That's the key innovation. But in terms of the ultimate radiation then of the angiosperms, there were several other concomitant traits: double fertilization, flower, and the fruit. All right, so here are the questions, okay, that I would like you to kind of go back and study. All right, we can go through these. If you want to hang on, after class, we can maybe work through these, but if not, I'm done. So we can then all join up on next Tuesday. I will mention that your quizzes, we're getting them available. We'll send out an email and a note on Slack when they're all available. So with that being said, thank you very much. We're at the end. Happy to answer questions if you have them.